Nervous system, brain. Main questions of the lecture. This slide demonstrates the increasing of size and complications of the structure of the brain during phylogenesis. You see the size of brain in fish, frog, snake, bat, cat, monkey, and human brain. The comparatively the biggest one and the most complicated. This slide demonstrates the increasing of size and complication of the structure of the human brain during embryogenesis. This is the point, comparative size of the brain on the 25th day of embryogenesis. 40 days, 50 days, 100 days, 5 months, 6 months. The surface is smooth, no gyri. The first gyri appeared on the 7th month, and the amount increased significantly, and there are all gyri available to the moment of delivery in newborns. This is a human brain of adult at sagittal section. Ventricles and canals of the brain stain in blue. Brain, parenchymal organ, like other organs of nervous system. It consists of stroma and parenchyma. Stroma represented by capsule around. Consists of 3 meninges. Dura mater, arachnoid and pia mater. Layers of loose connective tissue going in the deepness of the brain. Brain consists of brain stem, basal ganglia, large hemispheres, cerebellum. Brain stem consists of medulla oblongata, pons, midbrain, and intermediate brain consist of thalamus opticus and hypothalamic origin. This is a human brain, the view from below, the brain stem, hemispheres of cerebellum and brain, and these ventricles and canal stain in blue inside the brain. Brain consists of gray and white matter. Gray matter composed by neurons body and glial cells bodies. They are organized in 2 manners: nuclear type organization in brainstem, in basal ganglia, and screen type of organization, when neurons organized in layers, in screens, like in cerebellum cortex and brain cortex. And white matter consists of nerve fibers organized in bundles, pathways, which convey information between brain and spinal cord, as well as between different parts of the brain. This is the transverse section of medulla oblongata, and you can see nuclei, groups of neurons organized in nuclei, and this is a gray matter and white matter, the bundles of nerve fibers, conductive tracts. Nuclei can be divided into two groups: cranial nerves nuclei, sensory and motors, and switching nuclei like inferior olive, reticular formation in the middle. Numerous nuclei, up to 100 nuclei, and many pathways we will study in human anatomy course in details. In histology course we will describe you microscopic structure and organization of cerebellum cortex and brain cortex. Cerebellum is the center of equilibrium and coordination of movements. Cerebellum analyzes information from outside and coordinates movement and support equilibrium, acts through the motor neurons of the spinal cord. Cerebellum consists of two hemispheres and vermis between. It consists of nuclei in the deeper part and cortex, which cover the surface of hemispheres. There are gyri, like extensions, and sulci between, like folds, invaginations, both in cerebellum and in brain cortex. It significantly increases the surface and volume cerebellum cortex and brain cortex also. You can see that white matter make like the trunk in the middle and enter to the every gyrus and branching for smaller and smaller layers. And gray matter located outside, it covers the surface cortex. At low magnification you can see the white matter which enter inside every gyrus, and gray matter cover the surface from outside. Gray matter consists of three layers: the outer molecular layer, it's here, the middle ganglionic layer, and the inner is granular layer. At middle magnification in the gyrus you can see the white matter in the middle and gray matter outside, which consists of three layers: the outer layer molecular layer, the middle ganglionic layer, and inner granular layer. This is molecular layer, ganglionic layer, and granular layer at high magnification. You see that ganglionic layer consists of one layer of ganglionic cells or pear-shaped neurons or Purkinje cells. The dendrites branching, initially large branches, then smaller and smaller in molecular layer. Basket cell, its axon branching and produce the basket around bodies of ganglionic neurons, Purkinje neurons. And you can see also small, numerous granular cells. This is schematic drawing of cerebellar cortex. You see the two gyri in 3D, transverse section and longitudinal section. You see the molecular layer of gray matter outside. Ganglionic layer consists of Purkinje cells and deeper white matter. Purkinje neurons have dendrites starting from apical portions of cells, and they branch intensively in molecular layer, transverse plane concerning to gyri. At longitudinal sections, the same cells have narrow dendrites, trees. These dendrites, trees, have corresponding projections on the surface of gyri. In molecular layer, there are two types of neurons: small and large stellate neurons. The axons branching and produce synapses on dendrites and body of Purkinje cells. There is also basket cells staying in green. The axons going transverse to gyrus, and the axons branching and produce baskets around the perikaryones of Purkinje cells. In granular layer, there are two types of cells: red ones, numerous granular cells. They have short dendrites and long axon, which go into the molecular layer and branching for two parts, and these axons going in longitudinal direction concerning the gyrus, passing numerous dendrites, trees of Purkinje cells. How the information and the cerebellum cortex from the periphery? Through the afferent fibers. There are two types of them: mossy fibers and climbing fibers. Mossy fibers branching in granular layer and compose synapses on dendrites of granular neurons. And another type, climbing fibers, go into the ganglionic and molecular layer and convey information directly to Purkinje cells. And mossy fibers convey information initially to granular cells, and they convey this information through the axons to dendrites of Purkinje cells. This information treated by Purkinje cells, they analyze it, make the decision, and convey the commands to the other parts of the brain through the axons of Purkinje neurons. And these axons make synapses on the neurons of cerebellum nuclei. And cerebellum nuclei neurons convey information to the motor neurons of the spinal cords, and they convey information to the muscles. Brain cortex is the grey matter covering the brain hemispheres. Its thickness between 2 and 5 millimeters. It may gyri extensions and sulci invaginations, folds. It increases in 3 times the surface of brain cortex. There are a huge amount of neurons and much more glial cells in the brain cortex. They organize in screen-like manner. They produce the 6 layers in brain cortex. The first layer, the superficial layer, just under the meninges, which cover the brain. It's molecular layer. It contains few stellate cells and a lot of nerve fibers, bundles, going parallel to the surface of the brain. The second layer, called outer granular layer, it consists of numerous stellate neurons. The third is pyramidal layer. It consists of middle-sized pyramidal neurons. The inner granular layer consists of numerous stellate neurons. The fifth ganglionic or internal pyramidal layer consists of very big, very large pyramidal neurons. The sixth layer is multiform layer. It consists of small neurons of different shape. Cytoarchitecture, its specific organization of neuron, and myeloarchitecture is specific organization of nerve fibers, pathways. There are 50 fields in brain cortex. There's different function and structure. All regions, the grey matter consists of 6 layers, but their structure varied from field to field. There are 2 marginal types of brain cortex: a granular, with well-developed 3rd, 5th, and 6th layers. This structure typical for motor fields of brain cortex, like in pericentral gyrus. And granular type is well-developed 2nd and 4th granular layers. It's usually in sensory parts of brain cortex, central parts of analysis. The model or column of the brain cortex is functional and structural unit of the brain cortex. The temporal symbols of several hundreds neurons, including all 6 layers of cortex. It's really like columns, width's about 300 microns. It consists of 3 parts: the entrance through the afferent fibers, centrally located cortical fibers, which come in from another part of the brain cortex. they're mostly axons of pyramidal neurons from other columns, and 2 thalamocortical fibers, which bring information from thalamus opticus of intermediate brain. The zone of information treatment, several hundreds neurons which treat this information, and exit of commands through the axons of pyramidal neurons, stained in blue. There are about 3 million of such columns. This slide demonstrates the development of the brain cortex during embryogenesis. You see the layer of matrix cells, which mitotically divided intensively, and every minute appeared thousands of new future neurons, neuroblasts. And you also see the special glial cells, like trunks, and new developed neurons, neuroblasts, moving along them and find their specific place in future brain cortex. You see the layers, future layers. And this is dynamic of brain cortex development in embryogenesis. This picture demonstrates the development of the brain cortex after birth. This is the brain cortex in newborn, and 3-6 months after birth, 1-3 years, 5-6 years. The brain cortex thickness slowly increase and reach maximum to the 6 years. After that, it not increase significantly. The amount of neurons does not increase. It stable from delivery. It may slowly decrease during the life. Amount of gyri also will not change, but the brain cortex became more and more complicated because every neuron compose more and more interneuronal connections, and every neurons produce connections with these many thousands of other neurons of the brain cortex. Blood-brain barrier is the barrier between blood circulating the brain blood vessels and neurons of the brain. This barrier consists of endothelial cells, and by processes of astrocytes which cover both blood vessels and neuronal bodies. Together these satellite oligodendrocytes. Blood-brain barrier prevent the entrance of biologically active substance from blood into the brain. For example, biogenic amines, which produced by suprarenal glands medulla, which produce adrenaline and noradrenaline, they cannot pass this barrier because they destroyed by special enzyme monoamine oxidase. Unfortunately, alcohol, nicotine, narcotic drugs can easily part this barrier